So hello, everybody. We're back again to what we're reading this month. And this time we're with Lena Neuenkamp. And because she published this year a very nice paper on ecology letters that is entitled Light Availability and Light Demand on Plants Shape the Arbuscular Mycorrhizal Fungal Communities in the Roots. So please, Lena, tell us, what is this paper about?
Hi, Guille. Thanks for inviting me. This paper is about trying to understand what factors are actually shaping the AM fungal communities in plant roots. And because we can't really measure fungal traits and we can't really trace the fungi, or it's very difficult, we try to eliminate different factors which could drive the fungal community composition. And one idea is that it's the strategies of the plants, how to deal with light or light shortage, because light enables the plants to do photosynthesis and to produce carbon, which is the currency of the plant mycorrhizal interaction. And so we were thinking that having more or less carbon available would alter whether plants are more or less picky regarding their investment into mycorrhizal fungi. And this would finally shape how the fungal communities look like in plant roots.
So for your study, you compare communities that were shaded and communities that were open, right? And the diversity, the plant diversity of these communities were different. Do you think this might affect the pattern that you found?
Yes and no. On one hand, yes, because it's the idea that different plant communities have, like AM fungi are not really host specific. It's not there's one fungus pairing up with one plant, but there are certain host preferences because different plants have different requirements and different ability to invest into mycorrhizal fungi, and different AM fungi have different abilities to acquire nutrients and provide other services. So if I have different plants around me at the high diversity, I probably have a high diversity of different interaction niches, which enable home to a lot of different AM fungi. And if then I look at the plant growing in this community, this plant can select from this pool of fungi, and so the plant community or plant community diversity will indirectly affect what fungi are available for the focal plant to select the fungi from, or be selected by the fungus. We just don't know.
Yeah, very cool. This is related with one thing that I like from the, I mean, I like very much the paper, but one thing that I think is a really good idea, that you measure the AMF communities in the roots, but also in the soil, even though your question was focused on the root. Can you explain us a little bit more like that? Why you decided to also check the AMF communities in the soil?
I guess the one idea was that comparing roots and soils can be and the AM fungal community can be a way to detect host selectivity, because ideally, if we would like to select host selectivity, we would need to really look at the border of the root and really see the processes. How does it work that certain fungi get in and certain fungi don't? And this is hardly impossible. And so one idea is that if the soil promotes, like it has been found for plants, promotes a pool of AM fungal species, and the roots select a subset out of it, and so and if there would be no processes, there would be just a random exchange between fungi in the roots and fungi in the soil, we would find the similar patterns in response, for example, to different ecosystem conditions. in roots and in soil. And then we would say, okay, it's just a random, the roots take whatever is there in the soil. But as soon as we find any deviation in the roots compared to the soil, in respect to AM fungal communities, we can refer assembly processes. So it could be that the fungi, the plant selects for a small group of fungi, so it's a kind of a host specificity or a selection, or it selects for a higher diversity of fungi. So it could be that there's a lot of Glomeraceae fungi in the soil and maybe 4 other genera and families, but the plants always have all 5 genera. So there should be one active process leading to a high diversity of fungi in the roots. And then this you can see by comparing the community patterns of fungi in the soil and in the roots.
Yeah, this is very, this pool concept is very interesting. Did you find like the evidence for the pool, meaning that the soil contains all the species that were found in the roots?
Yeah, we did find, I think it's in the either in the figure 1 in the Venn diagrams of the paper or in the supplementary material, that essentially there was 2 or 3 or like not less than 10 fungal species which were we found in the roots but not in the soil. And there was more species we found in the soil than in the roots, which show that we captured, like which show that we found what we expected.
Mostly everything. Yeah, just a quick question here, because I was thinking that maybe some species are like spores, get into the plant in the soil and then temporarily disappear from the soil and then sporulate again. And so this could be these 10 species, right? Or even the detection of the pool of species is never perfect. So it depends on the amount of soil and everything. Yeah.
Exactly. This is related to this. I think in previous papers we sometimes had the problem that if you only take, because AM, it depends on the density of AM fungal material in the soil, and if it's not very dense and you take really this standard 250 micrograms of soil, then you sometimes don't see this pattern because and we think it's because you don't really get a representative sample of the soil. So we have done work that it's really good if you take a bigger amount of soil and maybe also sample different locations to capture the whole diversity of fungi in the soil.
In regarding this topic, in the last paragraph of the discussion, you have a very interesting hypothesis that is saying that if you have similar composition of AMF in the roots and in the soil, this might be a good indicator for optimal conditions for plants, which I find really, really exciting. Can you expand on it?
Nice, thanks. Well, I guess one idea is the one idea behind, and maybe this is also an idea which got shaped in myself through this paper, is that what is selectivity in AM fungal symbiosis? Is it really plants select for fungi, or like actively, or is it maybe that fungi select for plants, or is it that AM fungi are obligate symbionts and they will colonize everything what they can because they need plants, their plant hosts to survive. So the idea is that high diversity of fungi in a plant root is the default case, because if a plant root doesn't close the gates, there will be loads of different fungi. Maybe in some roots there will be one fungus dominant, but the idea is that maybe high diversity is not a view of selection for high diversity by the fungi, by the plant, but rather that it's the default case. If there's no mechanism installed in the plant to select for any fungi, there will be a lot of different fungi. But then the risk is that, but the risk is, and this can only afford a plant with enough carbon, so a plant that grows in optimal conditions. that if you let everyone in, there are cheaters among it. There's some very beneficial fungi, but some also which is a bit a waste of your carbon money. So if a plant doesn't make the effort to select, and then the composition or the community diversity of the fungi in the roots and the soil are the same, meaning there seems to be no filter, the root is not really a filter for selecting fungi, then it means that the plant doesn't need to select because it has enough carbon to spend on whoever fungus comes in, or has other, or can allow a high diversity of fungi, because the benefit might also be that some very useful fungi, but maybe expensive fungi are among it, who can protect you against pathogens. So, and because we found the other pattern that in case of not optimal conditions, so when shade intolerant plants really suffered in shade, and they likely could not produce enough carbon through photosynthesis, then they had a smaller selection of fungi in their roots compared to the soil. And this comparison of patterns from the soil to the roots also enabled us to take into account that the communities, as you said, of shaded and not shaded communities were different. But if we compare the soil and the root relatively to each other, then you can detect whether there is a deviation from the soil pattern and the roots, and then you can refer to community assembly.
Yeah, this is I find it quite exciting. Also, if you start thinking like this opens doors in terms of mycorrhizal multifunctionality. You mentioned pathogens in light conditions. I'm not sure, but I guess you have more proper conditions to produce more sugar. So do you think these conditions, or what do you think will be this multifunctionality of the AMF plays a role here with light and shade?
I think that in general, I agree that probably a higher diversity or genetic diversity of fungi will provide a higher chance that the fungi will provide different functions. Not completely different functions. I think it's just better. The compatibility with certain plants is just better. So some might be more, and I think a big part is also the adaptation to certain environments. So some might, so if you have a high diversity of fungi, you probably could think of the insurance hypothesis. You have probably some fungi which still can work. And in this case, it's very similar. I think the ideas I got, because my background is in plant ecology, is a lot from plant functional ecology, where the idea is that if plants have a high diversity of traits, this means that they can offer a lot of functions, and they can also react in a variety of ways to the environment. And then in all, it ends up in a higher productivity or in higher resilience of the plant community. And there we also work with comparison of a random plant assembly to the diversity of the traits, and whether there's a selection of traits to adapt to some environmental pressure, or there's a diversity of traits, which then indicates that being very multifunctional in terms of a plant community is very important. I guess in our experiment comes also the factor time, time in the sense of stability, because in this sense we would look at the graph. I think it's the bar graph of figure 2, where there's like stack bars showing the different families and the indicator species, and we would see like, okay, it seems then that the forests or the shaded ecosystems, which have a more even distribution of very abundant and important species and AM fungal families, are more multifunctional in terms of their fungal community than the open grasslands. I won't sign that, because we know that there's a high diversity of fungi, although it's within maybe the genus of Glomos, but a high diversity of fungi in grasslands, and it's highly has a strong correlation with plant communities, which is not the case in really in the forest ecosystems. So I guess on one hand, it might be that fungi in the forest cover a bit more of different functions, because it's just because the environment in forest is much more heterogeneous. One has more developed soils close to the trees, and then one has soils which are bare, or there's disturbance to bypores, which is disturbing the mycorrhizal networks. Whereas in grasslands, there's less disturbance. So I think it just is more time that really community structure can establish over time. And then it's like with plants and other communities, there's some species, and in this case it's Glomos, which become dominant, and others which are less dominant. And one needs to take into account as well that the variety of plants in terms of their mycorrhizal status and type are much more heterogeneous in forests than in grasslands. And so the tighter match of grassland plants being often obligately mycorrhizal, and then you have mycorrhizal fungi always available. I think it's maybe a bit too much to talk about co-adaptation, or co-evolution, but I think that the stable time plants and fungi are there over and not disturbed over a long time will eventually lead to an adaptation to each other, and it will be properly. There's certain fungi being more adapted to a certain plant and vice versa, and this creates this maybe the higher dominant structure of certain plants, certain fungi being more abundant, but also the plant being more selective for certain fungi, and maybe less flexible. And in the forest, the plants and the fungi are confronted with very heterogeneous conditions spatially, but also temporally. And then the plants it might be one consequence why we find more facultatively mycorrhizal plants in the forest. So they adapt to the situation that they don't know whether they can invest into mycorrhizal fungi or not, whether there's enough AM fungi available, especially if you grow in the neighbor of ectomycorrhizal trees. But on the other hand, it also makes the life for fungi more insecure, because they deal with plants who let them maybe in and who don't. So there's no time for creating this community structure so much.
So it's like the shaded, if I follow you, the shaded is kind of a less optimal or more variable or more heterogeneous for the establishment of this long-term or evolutionary even temporal. No, quite interesting.
And this could be also an idea. I think what I had in mind, that shade tolerant plants in this case are maybe more flexible in their choice. They are not relying on a certain fungi, because they've learned that it comes up whatever it comes up, and then of course they have optimized their carbon resource strategies. And you could also see it. I think there's one graph where we plotted the Ellenberg values from the plants to their fungal richness in the roots, and what one sees is that under shade, the most shade tolerant plants had the highest diversity of fungi, and this was exactly the pattern. And then with increasing Ellenberg numbers, so with increasing need for light for the plants, the the selectivity, the number, the richness of fungi in the roots decrease, which supports the idea of maybe being more selective. Yeah.
This lead me directly to the next question, that is from the figure 2, where you compare the different censuses. You did one census in October 2012, I think, and another census in June 2014. And then, very interestingly, so the first census it was just 1 year after the experiment was set, and then you compare the patterns, and the patterns change or evolve or are even opposite. And you were already talking about these assembly processes. Can you expand on it?
Yeah, I was a bit thinking about these processes because, of course, the experiment was not, the first census also included plants which were sampled not exactly at the same time point, but depending on when they germinated the first time. But we thought in this sense it's the most consistent in terms of when the plant has germinated and should develop. At this seedling stage we sample, and then we sample after the plant is a year or a year and a half old and has established. I guess one thing is that dynamics in very early seedlings are different than in more where we don't deal with adult plants, but with the older the plant grows. I think it different mechanisms shape the plant AM fungal interactions, because the seedlings partly have different sorts of roots, or they get some, I guess some seeds have some nutrient packages to support their seeds, and so maybe the roots don't need to be selective or don't need mycorrhizal so much, so it's maybe more random processes. And in terms of light and shade treatment, it might also be a bit, we thought a bit about it, about like a shock reaction. It's like, okay, I take what I get in terms of mycorrhizal fungi and light, and then by time it reaches an equilibrium with the seedling according to their characteristics. Here, shade tolerance can maintain over a long time, and so that's why we think that the patterns evolve rather like the patterns which we expect over a long time will be those which we see after the second census.
Cool. Just have a question about one. If I understand, one of the messages of this paper, or potential message, is that the shade tolerant plant species can host more generalist AMF compared to the shade intolerant species. Is that it?
Yeah, I think in a way it's true, because my view on it, I think, or the one take-home message is that it depends on the conditions, but because shade-tolerant plants in general seem to be less stressed by shade, obviously, because they're adapted to shade, and so they don't need to be selective for fungi. And so in this sense, they are maybe shade-tolerant plants are more generalist in terms of their AM fungal community, so they don't select that much. But shade intolerant plants do. Funnily, when we look at the indicator species analysis, they seem rather to select for very widespread AM fungi, so for glomos. So maybe it's more the perspective from the plant than the fungus about the generalist and specialist. Because due to the lower selectivity of the shade tolerant plants, they include a higher variety of fungi in their roots, which are fungi like the different families. It's Paraglomeraceae, and it's Diversisporaceae and Claridioglomeracea. It's not the right name, but it's complicated. Anyway, you can, and I think it's rather, at least I would, after this paper, I thought it's maybe really rather the plant who selects, and it's the plant who is more generalist in interaction, and they can be generalists when interacting with many fungi if they're in good conditions or have a good carbon strategy. Yeah.
Cool. It will be. I just thought it would be very interesting to add on top of this experiment some, I don't know, some pathogens and see whether the pattern change or not. It's really neat experiment. I really like it. Yeah, so I think you have covered the main take-home message, but do you want to rephrase it shortly or?
No, I think I'm.
It's covered. Okay.
Good, yeah.
So then I want to ask you. So, what are you working now? What is the topic you are developing now?
Of course, have big ideas after coming from plants and restoration to take mycorrhizal fungi to explain plant restoration success, and now to better understand mycorrhizal fungal communities. I think I was intrigued by understanding their relevance for ecosystem functioning, and so I'm working now in a study where I try to link soil biodiversity, including pathogenic fungi, mycorrhizal fungi, but also other trophic groups in the soil, to multifunctionality of not so northern territory, but now it's rather drylands in Spain and in other countries where they are occurring. But in this case, also to include plant traits, because based on the idea of niches, my idea was that if there's a plant community with high functional diversity in terms of resource strategies, but also in terms of root traits, there are more possibilities for interacting with fungi. And so if I have the higher possibility as a plant to interact with a higher variety, or a plant community with interacting with a higher variety of fungi, the chance that I have multiple functions covered is bigger. And so in the end, it should lead to a higher functioning of the plant community. And it has been found that higher functional diversity of plants is increasing functioning of the ecosystem. And my idea is whether this is not mediated actually by the microbes. And this is what I try to find out, to zoom a bit into functioning and into the functional strategies. Yeah.
Oh, very good. So thanks a lot for your time. It was a pleasure to talk to you, Lena.
Yeah, thanks for inviting. So exciting.
Bye.
Bye-bye.